Hello everyone, welcome to your channel Biology Spark. Today we will discuss regarding the regulation of spermatogenesis and structure of sperm cells. So one of the important topic of human reproduction in standard 12. Spermatogenesis is the formation of sperm cells, and which process or which things that regulate the spermatogenesis, formation of sperm cells. Then we discuss regarding the structure of sperm cells. So spermatogenesis, once primary spermatocyte leads to the formation of 4 spermatozoa, and we know that millions of sperm cells are produced. So hormonal regulation is that how the hormones regulate the spermatogenesis. So particularly the gonadotropin releasing hormone, which is secreted from hypothalamus. Hypothalamus leads to the secretion of gonadotropin releasing hormone. This gonadotropin releasing hormone stimulate the anterior pituitary gland for the release of gonadotropins hormone, that is FSH and LH. FSH, follicle stimulating hormone, LH, luteinizing hormone. So these both are called as a gonadotropin hormones. The gonadotropin releasing hormone leads to the secretion of this hormone from the anterior pituitary gland. Now, what are the function of FSH? This FSH entered into the testes and it stimulate, it act as a through the secondary messenger cells, it entered into the testes, seminiferous tubule, and it stimulate the Sertoli cells. So this FSH entered into the testes. Through the testes, it stimulate the Sertoli cells to produce androgen binding protein. This androgen binding protein stimulate this androgen binding protein, it stimulate the process of spermatogenesis, ultimately the formation of sperm cells, as well as it also increase the concentration of testosterone. The testosterone hormone is increased in the seminiferous tubule. So androgen binding protein play two major role. One, it leads to the spermatogenesis, and second one, increase the concentration of testosterone in the seminiferous tubule. Now another hormone, luteinizing hormone, that stimulate the Leydig cells, that is interstitial cells which are present between the seminiferous tubules to secrete testosterone hormone, that is also called as androgen. So this testosterone hormone is secreted from Leydig cells, but LH hormone stimulate the secretion. These testosterone hormones have a multiple role. Particularly, it increases the process or it leads to the sperm production. So this testosterone hormone is produced in interstitial space, but androgen binding protein allow this testosterone into the seminiferous tubule, because spermatogenesis is occur in seminiferous tubule. So this androgen binding protein increase, or either it leads to the entry of this testosterone from interstitial space into the seminiferous tubule. As well as it also responsible for the secondary effect, that is secondary sexual characters. It also gives a negative feedback to the hypothalamus as well as anterior pituitary gland in excess concentration. If this testosterone is released in excess, then it have a negative feedback to the hormones LH, ultimately anterior pituitary gland as well as hypothalamus. It inhibit both. So no testosterone, no spermatogenesis is there. So if excess spermatogenesis is there, then this testosterone itself inhibit the cycle. Another negative feedback is also given by Sertoli cells. This, if there is an excess amount of spermatogenesis, in excess spermatogenesis, Sertoli cells produce inhibin hormones. So it produces inhibin hormone. This inhibin hormone again gives a negative feedback to the anterior pituitary gland, mainly to the anterior pituitary gland, and to some extent it also have a negative effect on hypothalamus, and it result in the suppression of synthesis and release of FSH. So ultimately no FSH, no androgen binding protein, no spermatogenesis. So negative regulations is there, same cycle is there. So gonadotropin releasing hormone act by pulsatile release. FSH is a follicle stimulating hormone which sensitize the Sertoli cells to release testosterone. It also promotes the secretion of inhibin, but inhibin is a negative feedback. And androgen binding protein from the Sertoli cells, it keeps the high level of testosterone within the gonad. So within the seminiferous tubule, it maintains the high concentration of testosterone. LH hormone, luteinizing hormone, it stimulates the Leydig cells. Positive means stimulate, negative means decrease. So inhibition. So positive effect over the Leydig cells which increase the secretion of testosterone by the activity of the enzyme cholesterol desmolase enzyme. So testosterone, again this androgen binding protein which ultimately increase the permeability of testosterone into the seminiferous tubule, and this testosterone also regulate FSH and LH level by negative feedback inhibition. So inhibin also regulate the FSH as well as LH and testosterone. So these two hormones, testosterone and inhibin, both regulate the amount of FSH and LH by negative feedback inhibitions. And testosterone stimulates the spermatogenesis. It itself also stimulates Sertoli cells, and ultimately it stimulates the process of spermatogenesis. So this one is the hormonal control over the process of spermatogenesis. मतलब sperm cells का जो amounts है वो ज़्यादा भी नहीं रहता और कम भी नहीं रहता due to this hormonal mechanism. अगर more amounts में sperm cells formed हो गए तो testosterone itself inhibit the formation of sperm cell. Now structure of sperm cells. So human sperm cells is about 70 micrometer low. उनकी साइज 70 माइक्रोमीटर है. स्पर्म सेल्स के 3 पार्ट है, हेड, मिडल पीस एंड लॉन्ग पार्ट जिनको हम टेल बोलेंगे. सो हेड साइज इज अराउंड 4 to 5 माइक्रोमीटर. middle piece that is 4 to 5 micrometer, but maximum length is of 55 micrometer, that is tail. The each part have a specific function. The nucleus which is present in the head region and it contains the 23 chromosomes. So heredity material is present in head region. The head region also have a crown like structure, cape like structure, that is acrosome. The middle piece particularly rich in the mitochondria, so it generates the energy for the motility, and the sperm cells which is also called as a flagellum, and that provide motility for the propagation of sperm cell. So it is a haploid gametes around 60 micrometer in length, and it contains head, middle piece and tail. The head of sperm is narrow shape and which have a large nucleus which is haploid in nature. 23 number of chromosomes are present. Anteriorly, the nucleus is covered by acrosome. This type of structure, cape-like structure is present over the head region, that is acrosome. You have to remember here, the acrosome is formed by Golgi complex. So acrosome formation is occur from the Golgi complex, and it contains the Leydig enzyme to dissolve the egg membrane during the fertilization. So the Leydig type of enzymes are present. You have to remember the enzyme that is also called as lysine, sperm lysine, as well as it also have a high luronidase type of enzyme. High luronidase enzyme which dissolve the egg membrane and easily the penetration of sperm cells. So acrosome help for the penetration of sperm in the egg cell at the time of fertilization. Now neck region. The neck is a short and slightly constricted segment which is made up of projections located between the base of head and the middle piece. Neck region short है, हेड रीजन और मिडल पीस के बीच में प्रेजेंट है, बहुत छोटा सा पार्ट. इट कंसिस्ट ऑफ सेंट्रियोल. विच थिंग्स आर प्रेजेंट इन नेक, तो नेक रीजन में सेंट्रियोल है, दो सेंट्रियोल है, बोथ आर एट राइट एंगल टू ईच अदर, उनको प्रोक्सिमल एंड डिस्टल सेंट्रियोल बोलते हैं। नेक रीजन में प्रोक्सिमल और डिस्टल सेंट्रियोल प्रेजेंट है। What are the function of both centriole? The distal centriole forms and give attachment to the axial filament of sperm tip. So this centriole already it serve as a basal body for the formation of axial filaments. This one is called as a axial filaments, which is present up to the end of tip. So ये distal centriole है, वो axial filaments को अटैचमेंट प्रोवाइड करता है फ्रॉम नेक रीजन से। बट जो प्रोक्सिमल सेंट्रियोल है, एक्चुअली इट डजंट हैव पर्टिकुलर रोल इन द स्पर्म सेल्स, बट it have a potential activist within a egg cell during the first cleavage of fertilized egg cell. हमें यहाँ पे याद रखना है कि एग सेल के पास सेंट्रियोल नहीं होता, तो जो भी जाइगोट में सेंट्रियोल है वो प्रोक्सिमल सेंट्रियोल है, और वो सेंट्रियोल एक्टिव एट द टाइम ऑफ फर्स्ट क्लीवेज आफ्टर फर्टिलाइजेशन। तो जाइगोट का जो डिवीजन है वो प्रोक्सिमल सेंट्रियोल कंट्रोल करता है। We know the function of centriole. Centriole help in the process of cell division. So this both centriole are present in the neck region, proximal and distal centriole. The middle piece. The middle piece is also called as energy chamber. It is considered as energy chamber of sperm cell. It is surrounded by spirally arranged mitochondria. It is surrounded by spirally arranged mitochondria like this around the centriole or axial filaments. Around these axial filaments, the spirally arranged mitochondria are present in the middle piece, and entire sperm is enclosed by the plasma membrane. So this entire sperm is enclosed by the plasma membrane, the outer covering. The ring centriole, the function is unknown, which is also present at the distal end of middle piece here. The dot-like structure are ring centriole, but the function is unknown for this centriole, which is present at the distal, means end of middle piece. So this middle piece have a mitochondria and which serve as an energy chamber. It leads to the generation of energy, and that energy is utilized for the motility of sperm cell. Move to the tail. The sperm tail is a thin, elongated, and around 80% of entire length of sperm cells. It forms around 80% part of sperm. The axial filaments are present in the tail region. You have to remember the 9+2 arrangement is there, because it is a flagellum, so it have a 9+2 arrangement of filaments, microtubules. So axial filament complex, which is extend through the most part of tail, end तक ये फिलामेंट्स एक्सटेंड हुआ है। 9+2 पैटर्न है माइक्रोट्यूब्यूल्स की जो अरेंजमेंट है, और वो ओरिजिनेटेड होता है बेसल बॉडी ऑफ सेंट्रियोल, विच आर प्रेजेंट इन द नेक रीजन, दैट इज डिस्टल सेंट्रियोल। 9+2, that is 9 pair doublets are arranged in a circle around the central pair, around the two isolated microtubules which are present in the central. So that's why it is called as a 9+2 arrangement. That axial filament pass from middle piece and most of the tail, and particularly it is responsible for the motility of sperm. So these are the various part of sperm. Each part have a specific function. Head region have a nucleus, acrosome. Acrosome help in the penetration. Nucleus is a contain heredity information. Neck region have a centriole, which help in the formation of this axial filament, as well as also it serve as a cleavage of fertilized egg cell. Middle piece which have a mitochondria, so powerhouse, again energy chamber, and tail that have a axial filaments responsible for the motility of sperm cells. So this is the hormonal regulation as well as structure of sperm cells. Okay, so hope you enjoy this video, and don't forget to like, share and subscribe.